Hello dear students, welcome to the video lecture series on Western blotting under Unit 4A Immunoblotting Techniques from the syllabus Pharmacy Council of India, 3rd year B Pharmacy, 6th semester, subject Pharmaceutical Biotechnology, subject code is BP605T. Myself Dr. J P Lavande, Associate Professor, Department of Pharmaceutics, Shivlingeshwar College of Pharmacy, Almala, Taluka Ausa, District Latur. So as far as the Western blotting is concerned, in previous lectures we had covered principal SDS page, Western blotting and its detection in the form of 5 different steps: transfer blocking, primary antibody incubation, secondary antibody incubation and analysis. In this particular video, we will go through the Western blotting diagram and the detailed protocol associated with the Western blotting. From study point of view, we will cover the first 3 protocol steps in this particular video lecture and remaining, that is step 4 to step 9, in the subsequent videos. So the Western blotting, that will consist of, just from summary point of view, it involves the sample preparation associated with the protein extraction, protein assay, running the proteins through the gel electrophoresis, staining the gel, then protein transfer to a membrane, then blocking, washing, incubating with the primary antibody, followed by incubation with the secondary antibody, then chromogenic detection or the chemiluminescence detection. So the whole steps that will go through the 9 steps. In that, the step 1, that is associated with the sample preparation or protein extraction, which is associated with preparation of proteins from cell culture, preparation of proteins from tissues and the protein assay. Sample preparation associated with the preparation of proteins from cell culture. Culture cells in a tube until the density is up to 80%. Digest cells with 0.05% trypsin or scrap adherent cells of the tube using a cold plastic cell scraper. Wash cells with pre-cooling buffer system, that is PBS, for 2 times. Centrifuge at 3000 rpm for 2 to 3 minutes and remove supernatant. Then gently transfer the cell precipitation into a pre-cooled tube. Add mammal cell protein extraction reagent, which is 5 to 7 times the volume of the precipitation, into the tube. Blow and beat repeatedly. Using ultrasonic processor to break up if the solution is murk, ultrasonic processes for 2 seconds and pause for 2 seconds. The power is 100 to 120 watt. And turn off the power when the murk solution becomes clear. Then lactate the solution on ice for 4 to 5 hours. Ultrasonic processes the solution again. Centrifuge at 10,000 rpm for 10 minutes. Discard liquid on the top and lower cell debris and take out protein solution in the middle. Mix the protein solution with booster SDS PAGE loading buffer 2 times in the same volume or with SDS page loading buffer 5 times in volume ratio of 4:1 evenly, and then denature the solution in 100-degree water bath for 5 minutes. The solution could be used immediately or early curted for store at 20 degrees Celsius for several months and at 4 degrees Celsius for 1 to 2 weeks. Preparation of the proteins from tissue. Place surgically resected tissues in pre-cooling normal saline immediately. Wash away blood on the tissue. Chop the tissue into several small pieces, 0.1 to 1 gram after weighing. Add booster enzyme inhibitor cocktail prior to the homogenate and add 10 ml mammal tissue protein extraction reagent per 1 gram of tissue. Grind the tissue until fully homogenized. Lactate tissue homogenate on ice for 4 to 5 hours after treated by lateral sonic processor. Centrifugate at 10,000 rpm for 10 minutes and take out protein solution in the middle. Mix the protein solution with booster SDS PAG loading buffer 2 times in the same volume or with SDS PAG loading buffer 5 times in the volume of ratio of 4:1 evenly, and then denature the solution in 100 ml water bath for 5 minutes. Protein assay. After protein sample collection, concentration of proteins should be quantified to ensure equivalent loading amount of each protein. The most commonly used assay kits are Booster BCA protein assay kit, Micro BCA protein assay kit, and Comasi Plus protein assay kit respectively. Comasi Plus protein assay is easy to carry out and to consume less sample. However, different proteins perform with great variety and poor standard curve linearity will be generated. pH value may change under the function of a variety of elements such as high concentration of tris ADTA, urea, glycerol, sucrose, acetone, ammonium sulfate, and detergent. And this will subsequently influence color development and interfere protein quantification. BCA protein assay is valued by its stronger stability, higher sensitivity, higher flexibility, and colored complex form is more stable with little interfering substance. BCA protein assay has an obvious advantage over Comasi Plus protein assay that is not influenced by the detergent. Step 2. Preparation of gel. Prepare resolving gel in proportion. Blend solution gently to avoid involving too much oxygen. Pour gel into 2-layer glass pane in gel aperture slowly. Add a sheet of water to prevent inhalation of oxygen. Placing horizontally, incubate at 37 degrees Celsius for 60 minutes till gel solution is fully polymerized and concentrated. Prepare stacking gel in proportion. Blend solution gently. Absorb water on concentrated resolving gel with filter. Pour stacking gel into 2-layer glass pane in gel aperture slowly. Insert comb into the gel carefully. Bubble is prohibited between comb tooth. Incubate at 37 degrees Celsius for 60 minutes till gel solution is fully polymerized and concentrated, and take out the comb gently. Note: Booster SDS PAG gel preparation kit is recommended for preparation of gel. Step 3. Loading the samples. Add booster loading buffer into electrophoresis chamber to a level above sample loading well between 2-layer glass pane. Ensure bottom of the gel is immersed in the loading buffer. Liquid level inside the 2-layer glass pane is higher than outside level. And then load samples which include treated sample, tissue, and cell sample protein for positive control. Recombinant protein for positive control and marker in wells. Load 30 to 50 microgram protein to each well and the total loading volume is less than 30 microliter. Note: the time should be short as much as possible to prevent diffusion of sample.